Hello students, welcome to Trunkie Academy. In last some classes, in past some classes, we study about the biotechnology, the different principles and the processes involved in the biotechnology, applications of biotechnology in agriculture and in medicine. Okay. Then we studied about the structure of DNA. Yesterday's class. Okay. Now today we will study about the search for genetic material. Like I told you yesterday, the DNA is the genetic material, deoxyribonucleic acid. Okay, it is a polymer of nucleotide, and now when the scientists were researching about the transmission of characters from one generation to another, okay, one generation to other, they were thinking of what may be the genetic material. Which is the genetic material that is present in the cell that is playing a role of transmission of characters from one generation to other. Okay, so they were on the search for genetic materials. Okay, and during this search for genetic materials, they carried out so many different experiments. Okay, and many scientists they experiment in different ways so as to understand what is genetic material actually. Okay, now. Which one? Okay, is it the RNA or it is the DNA or it is something else? Okay, so Gregor Johann Mendel, one of the very important scientists who did some experiments, is a pioneer in genetics. He did some breedings in the pea plant and came out with some very outstanding, important conclusion. Okay, and he formulated some laws like law of segregation, a law of independent assortment, okay, law of dominance. Okay. Walter Sutton, Sutton and Boger together, okay, they also did some experiment on with regard to genetics. Thomas Hunt Morgan, he also worked with Drosophila melanogaster, a fruit fly, okay, and studied about some important characters being transferred from the parents to offspring and the mode of transmission, and also trying to understand about knowing about the genetic material. Okay, and so many scientists they researched. Okay, so they were on the search of genetic material that is present. Okay, so they have understood that this genetic material is present in the nucleus. This genetic material is in the nucleus, and this is in the chromosome. Okay, this material is in the chromosome and chromosome is in the nucleus. That much they knew, but they had to know further than this. Okay, so there was some many experiments were conducted by different scientists, and they were able to understand later on with the help of the work done by the different scientists, and thus the Mendel and T.H. Morgan, etc. Now, transforming principle. This is one of the very important things. Okay. Based on this principle, we can have some idea, or you can say we have a clear idea that this is the genetic material. Transformation experiments of Griffith. He is one of the famous scientists who did some experiment. Okay, and he then concluded which is the genetic material. Okay. Now, transformation experiment was first performed by a British bacteriologist Frederick Griffith in 1928. Okay. So this is why it's called, that is the reason it is called the Griffith's experiment. He carried a series of experiments with bacteria, okay, and that we'll see in a while, okay, what experiment he did and how he came to a conclusion that DNA is the genetic material, okay. There is one important famous bacteria on which this Frederick Griffith did experiment. The name of this bacteria is Streptococcus pneumonia, okay, or Streptococcus pneumoniae. Now, I have already explained about the structure of the bacteria. You already studied in class 11. The coccus, the spherical bacteria are the coccus. It's Streptococcus, So, Streptococcus pneumonia or pneumococcus, also called pneumococcus, which is also known as Diplococcus pneumonia. So, on this bacteria, he did experiment. Okay, this was the organism for experiment. Okay, this parasite causes pneumonia in mammals, including humans. This Streptococcus pneumonia or pneumococcus are also called as the Diplococcus pneumonia. They cause pneumonia in this is called pneumonia in the mammals, including humans also. Okay, so this is the structure, the purple one. This is the violet one. This is the pneumococcus. Okay. Griffith's study, he studied about two strains in the bacterium. Okay, while studying about this pneumococcus, he found two strains, two varieties of this pneumococcus, and that was S strain or virulent, that is virulent, that is causing the disease, and another was which causes a covering of mucilage, smooth or regular colony, and the ability to cause the disease of pneumonia. Okay. And another was the R strain or non-virulent, which lacked mucilage sheath covering, forms the rough or irregular colony and is unable to cause disease. So basically there are two varieties, two types, two strains he found in this bacteria, pneumococcus, and S strain and the R strain. S strain being the virulent because it has a covering of mucilage, okay, mucilage covering. Whereas in the case of R strain or non-virulent type, there was no mucilage covering. Okay, so this R strain was not able to cause the disease. The S strain was causing the disease of pneumonia. A series of experiments were conducted by Griffiths. Okay, and then he was able to find the DNA as genetic material. So first, what he did is he injected the S strain into mice. The mice developed pneumonia and died, leaving its S strain cells recovered from the body of dead mice. Okay, so S strain is one. So what he did is first he injected the S strain into the mice, and that mice died because of the disease of pneumonia. And then, while collecting the strains of the bacteria, S strains were found from the body of the mice. Okay. So pneumonia and they died, and then what he did is the second stage, injected R strain into the mice. The mice did not get the disease and they survived. Living R cells were recovered from the body of mice. Likewise, in case of the first stage, what he did is injected the S strain, and S strain was recovered from the body of the dead mice. And in the second experiment, he injected the R strain, and the mice did not get the disease and did not die. Okay, and but similarly, because R strain was injected, so living R cells were found recovered from the body of the mice. Okay, so you see, so it was not dead; it was alive and survived, and R cells or R strain were recovered from the body of the mice in second experiment. Okay, and second part of the experiment. Now, Griffith was able to kill bacteria by heating them. Okay, what he did is he killed the bacteria by heat, so heating them. So he observed that the heat killed S bacteria or S strain bacteria injected into the mice did not kill them. So what he did is he killed the bacteria of S strain, and with that killed S strain bacteria that were injected into the mice, and that killed bacteria did not cause the disease, and the mice did not get the disease and did not die also. Okay? When he injected a mixture of heat killed, that means the S strain killed and the live R bacteria, the mice died. Now he was able. Now this was the pinpoint where he was compelled to think over. Okay? When he was injecting the heat killed S strain, the virulent one, okay, the mice survived. The pneumonia did not occur. But when he injected the mixture of heat killed S strain, the virulent, along with the R strain, mice died. So moreover, he recovered the living S bacteria from the dead mice. So he found that while recovering the bacteria from the dead mice body, he found the S strain of bacteria, S strain, not R strain. Okay. So injected heat kill strains into mice. The mice did not get disease and remained alive. Okay. but S or R strains are not found in the body of mice. So when he injected with the heat kill S strain, okay, mice survived, did not get the disease, pneumonia, and it remained alive. But when recovering the bacterial cells from the body of the mice, there was no R strain or S strain were found. Okay, so heat killed S strain, mice survived, and now injected heat killed S strain along with live R strain. Some mice suffered from pneumonia and died. Like I told you earlier, the living strain S strain cells found in the body of the dead mice. So that was his the important the outcome of the experiment, that when heat kill S strain is injected with the live R strain, which is non-violent, R strain is non-violent and not causing the, but mice developed pneumonia, suffered from the pneumonia and died. And while recovering the strain of the bacteria, S strain was recovered from the body of the dead mice. Okay, so heat killed S, and then pneumonia, it died, and now it is concluded that R strain bacteria had somehow been transformed by the heat killed S strain. So then he was thinking that R strain, which is non-violent, alive, okay, that somehow been transformed by heat killed S strain, which is, though it is heat killed, but the presence of that strain has somehow caused the transformation in the R strain to make it virulent. Okay, so some transforming principle is playing a key role in changing that R strain into S strain. Okay, so some transforming principle transferred from the heat killed S strain had enabled the R strain to synthesize a smooth polysaccharide coat and to make this R strain as virulent to cause the pneumonia and disease. Right? So that was his, the thought process started, and he started thinking about the transforming principle. Thus, must be due to transfer of the genetic material. So now he was thinking, what was that? What was that transferring principle? Why this R strain, which is non-violent, is not causing the pneumonia, has been transferred into virulent, okay, in the presence of heat killed S strain. So that may be, then it must be the genetic material. This may be due to the transfer of genetic material, okay. So however, the biochemical nature of genetic material was not defined from his experiment. Although he was, able to explain the DNA as a genetic material. What was the material that is transferring? Okay, that he was able to conclude from his experiment, but the biochemical nature, his structure in detail was not defined from his experiment. Okay, it was an indirect finding of the genetic material. Okay, from his experiment. The biochemical characters of transferring principle. Prior to work of Oswald Avery, Colin MacLeod, and Maclean McCarthy, they were the scientists who worked on the genetic material. The genetic material thought to be a protein. Okay, so after like we observed this in case of the Frederick Griffith. Okay, these scientists, Oswald Avery, Colin MacLeod, and Maclean McCarthy, they did some work, experiments, research on genetic material. Okay, so then till they worked on this and found genetic material is not protein. Before this experiment of these scientists, the genetic material was thought to be a protein. Okay, but which was not the correct. Okay, so they are the scientists: Oswald Avery, Colin MacLeod, Maclean McCarthy. They are the three scientists who worked and found that genetic material is not protein. So before the experiment of these gentlemen, it was the genetic material was thought to be as protein. Okay, so now after this experiment of these scientists worked to determine the biochemical nature of the transforming principle in the Griffith experiment. So these scientists, okay, they started thinking and also wanted to know what is that transforming principle which we were found, and and concluded by the scientists of, the Griffith experiment. Okay, so they purified biochemical proteins, DNA, RNA, etc. from the heat-killed S cells to see which ones could transform live R cells into S cells. So they purified, okay, and they discovered that the DNA alone from S bacteria caused R bacteria to become transferred. Now, after purification and after experimentation, they found that the only DNA alone, not the RNA or other part of the chromosome, only DNA is able to alone is able to change or transform the R bacteria into the vibrant form, or you can say the form of coating, like it is found in the case of the S bacteria. They also discovered that the proteins digesting enzymes, proteases and RNA digesting enzyme, RNAs, did not affect transformation at all. So the transforming substance suggesting that the DNA caused the transformation. So from this work, from these experiments, okay, from these observations, they found that it is not the enzyme, RNAs or proteases, okay, or other part of the chromosomes. It is the DNA that has caused the transformation of R strain into the S strain. Okay, they concluded that DNA is the hereditary material, genetic material, but not at all biologists were convinced. Okay, but after that, the other biologists, the scientists, were not convinced. Okay, how is that possible? Okay, how this DNA can be the hereditary material? They were, since the technology were not so advanced at that time. Okay, and there were some proof. Some there were indirect kind of evidences that are indicating that this hereditary material is DNA, but there were some of the scientists not convinced. The genetic material is DNA. Harse and Chase experiment is another experiment which also gave a new, you can say, the light on the understanding the genetic material. So what they did, Harse and Chase, the unequivocal proof that DNA genetic material came from the experiment of Alfred Harse and Martha Chase. These two scientists, in 1952, they did experiments, and experiment of these two scientists then confirmed, gave some satisfactory and understandable answer for that the DNA is the genetic material. Okay, so what did they do is they work with viruses. Okay, that infect the bacteriophage. Okay, so there is a kind of virus, they infect the bacteria, and these viruses are called bacteriophage. Okay, so the bacteriophage attaches to the bacterium and genetic material then enters the bacterial system. What happens? They have got some kind of structure at the base. They attach at the surface of the bacteria, and then slowly they release, they enter into the bacterial cells. Okay, and the bacterial cell treats the viral genetic material as if it was its own and subsequently manufactures more virus particles. So bacterial cells, okay, take the genetic material from virus and treats that viral genetic material its own and starts the producing more virus particles. Okay, they try to observe which component of bacteriophage, that is either the protein or DNA, entered the bacterial cells and directed the reproduction of the virus. Okay, so they wanted to know, because when it's attached, okay, some part of the virus that goes into the bacteria, okay, not so which is going inside. Okay, they wanted to know, is it this protein or DNA that entered the bacterial cell? Okay, Harse and Chase grew viruses on a medium that contained radioactive phosphorus, okay, a P32, isotope P32. And some others on a medium that contain radioactive sulfur as 35. So they are the isotopes of the sulfur and phosphorus, and they did some kind of experiment with these grew viruses, okay, with radioactive phosphorus and radioactive sulfur. Now, what they found is viruses grown in the presence of radioactive phosphorus contain radioactive DNA, because DNA contains phosphorus, but protein does not contain phosphorus. Okay, sulfur is found in the protein. So what happens in sulfur? Okay, now that protein. Now try to understand this here. In case of this, so what happens that the radioactive, the phosphorus that will go into the DNA and radioactive sulfur will go into the protein part. Okay, so the virus is grown in the presence of radioactive phosphorus and contain the radioactive DNA because DNA contains phosphorus but not protein does not contain this. Right? Now, radioactive phages were allowed to attach to Escherichia coli bacteria, and here you can see this. Then, as the infection proceeded, the viral coats were removed from the bacteria by injecting them in a blend. This is a kind of a device in the blender, and the virus coats were removed. Okay. So what happened? The radioactive phages were allowed to attach to the Escherichia coli. Okay. Then the infection, as the infection proceeded, okay, the viral coats were removed from the bacteria by injecting the blender. The virus particles are separated from the bacteria by spinning them in a centrifuge, spinning at a very high speed. Then, as the infection proceeded, viral coats were removed from the bacteria by injecting them in a blender. And then, what they found is, like, see, how they are attached right on the surface of the bacteria, right? So, bacteria which were infected with the virus, they had radioactive DNA, they are radioactive, indicating that DNA was a genetic material that passed from virus to bacteria. So bacteria were infected with the virus that had radioactive DNA, were radioactive. So even in the bacteria also, the radioactive DNA was found, indicating that genetic material that passed from the virus to the bacteria was the DNA. Bacteria that were infected with virus that had radioactive proteins were not radioactive. So bacteria that were infected with the virus that no radioactive proteins were found. So that means protein did not enter. Protein is not the genetic material. It did not enter. Okay, so radioactive sulfur was not found. So radioactive the sulfur is present. And this radioactive sulfur was not found. So this indicated that protein did not enter the bacteria from the virus. So therefore, genetic material is the DNA that entered from virus to bacteria. So this was the conclusion drawn from the experiment. So they were able to understand, okay, that we have introduced the radioactive phosphorus as well as the radioactive sulfur. They grew the virus into the medium where there was the radioactive phosphorus as well as the radioactive sulfur. Okay, sulfur being the part of the protein, it is integrated in the protein, and phosphorus being part of the DNA, it went into the DNA to be integrated. And when the experiment was conducted, attached on the surface of the bacteria, when DNA entered into the bacterial cell, and when they observed the DNA of the bacteria, they found that the radioactive phosphorus was there, but not radioactive sulfur was there, indicating that the radioactive sulfur did not enter. This entered did not means protein did not enter. Okay, so protein is not the genetic material, but genetic material is the DNA. It has entered into the bacterial cell or bacterial body, right? So we will quickly go through some experiments. Transformation experiment was performed by Kornberg, no, G. Gregaton, Mendel, no, Frederick Griffith. Yes, it is the Frederick Griffith. He was the scientist who did the experiment on the virulent and non-virulent strain of bacteria called pneumococcus or diplococcus or streptococcus pneumoniae. Okay, or streptococcus pneumoniae. Now the next is the pneumonia is caused by Staphylococcus typhi, no, Streptococcus pneumonia, or influenza, or retrovirus. It is the number 2, Streptococcus pneumonia, right? Genetic material is carbohydrate, DNA, fat or lipid, it is the DNA, very simple. This kind of question will not come, but just to revising it, we are just going through these questions. Okay. Now the properties. Now after the experiment of Frederick Griffith and Hassa and Chase, okay, now it was understood that DNA is the genetic material. Now what is the property of this genetic DNA? Okay, from the foregoing discussion, it is clear that the debate between the protein versus DNA as the genetic material was unequivocally resolved by Hassa and Chase experiment. Okay, so there was some kind of doubt. Okay, after the experiment of the Frederick Griffith in the minds of some scientists, that was very clear after the experiment of Hassa and Chase. Okay, whether protein is the genetic material or it is the DNA. So after the experiment of this Hassa and Chase, it was very clear that this is the how do you call this? DNA is the genetic material, right? It became an established fact that it is the DNA that acts as genetic material, not the protein that acts as genetic material, right? However, it subsequently became clear that in some viruses, RNA is genetic material. Yes, it is very few. In all the living organisms, okay, almost all the living organisms, it is DNA only, except some viruses. So all living organisms consist of DNA as genetic material, but except some viruses, RNA is the genetic material. So that is the tobacco mosaic virus, Q-bacteriophyte, HIV, etc. They are called the retrovirus. Okay, so retrovirus, or it is found in the in case of the tobacco mosaic, Q-bacteriophyte, HIV, etc. Okay, so a molecule that acts as genetic material must fulfill the following criteria. Yeah, then that is it. Now this is the important point. Okay, I am going to example also. If genetic DNA is the genetic material, okay, and it is confirmed, so this should have this kind of the feature, property, okay, it must fulfill these criteria, okay, to as being a genetic material. It should be able to generate its replication, because it has to transfer the information from that generation, parent generation, to the next generation, right? It should be chemically and structurally stable, and also it should provide a scope of slow changes in mutation that are required for evolution. Yes. So if there is change in the gene, in the sequence of the DNA, okay, in the base pair change, mutation, okay, so that brings the changes, okay, and even also by recombination of the different the bases pairs, that also is responsible for bringing the variation in the next generation. Okay, it should be able to express itself in the form of Mendelian characters. Okay, like in the Mendel's experiments we found. So it should be able to express itself. Like, okay, so these are the criteria for the one which acts as the genetic material. Okay, so these are the now. Because of the rule of base pairing and complementary, both the nucleic acids and the DNA and RNA have the ability to direct their duplications. Okay, so rule of base pairing and complementary. So there is the two strands in the DNA are there, like you know very well. Okay, the two strands. Okay, and these two strands have the complementary base pairs. Okay, their pairs are one is complementary to another. So knowing the bases in the one strand, single strand, we can predict the strand, the base on the another strand. Okay, so they have the ability to write the duplications. The other molecule in living systems, such as protein, fail to fulfill this first criteria itself. Right? This one. It should be able to generate its replica. So this is not done by the protein. Okay. Now. RNA is not a predominant and very suitable genetic material because why RNA is highly reactive and unstable. Like DNA is stable, but RNA is unstable, highly unstable. Now, two OH group present in every nucleotide in RNA is a reactive group and makes it liable and easy degradable. So that is another point. Okay, this presence of this group in this nucleotide and of RNA makes it reactive group and liable and easily degradable. RNA is also known to be catalytic as its function as enzymes, therefore reactive and unstable. So it is known to be catalytic. So it is we know that RNA also act as catalytic, show some catalytic action or function. It acts as a catalyst and function as enzyme, therefore it is reactive and unstable. Uracil present in the RNA is less stable as compared to thymine methyl uracil of DNA. So these are the points from which you can say that RNA is not a suitable genetic material because it's unstable and very reactive. RNA being unstable mutates at a faster rate. Okay, so in case of the evolution, we see that the change is not so fast. Okay, consequently, virus having RNA genome having a shorter life span, mutate and evolve faster, which is not in the case of other living organisms, right? So it is a slower. DNA is a predominant genetic material of the organisms because DNA is chemically less reactive and structurally more stable. Okay, it is less reactive and more stable, and DNA stability can be clearly evident in Griffith's transforming principle. Okay, so it is just transfer the and we saw that there is no much mutational change. Heat which killed the bacteria at least did not destroy some of the protein. Yes, this is a very interesting point. Try to understand this. Now this is the point which is that DNA stability can be clearly evident in Griffith's transforming principle. How? Because we know that by heating the bacteria were killed, but DNA was not destroyed. That means this DNA is stable to some extent, right? So heat which killed the bacteria at least did not destroy some of the properties of genetic material. That is the reason it was able to convert that R strain into S strain. This now can easily be explained in the light of DNA. The two strands being complementary if separated by heating, okay, come together when appropriate conditions are provided. Okay, when heating they are broken, denatured. As the temperature goes down, it becomes normal. After heating, they join. Okay, when the appropriate condition is provided, right? So Griffith's experiment showed that when the mice are injected with heat-killed S virulent along with the live R strain non-virulent, the mouse dies, and the strain obtained from the this mice, dead mouse, was the S strain. So that R strain was transformed into virulent S strain by the DNA that was present in the heat-killed S strain. Okay. Presence of thymine in DNA instead of uracil provides stability of the DNA. So, like in RNA, it's uracil. Okay, so that's unstable. But in case of DNA, we find the thymine and more stable. Okay, uracil in RNA and thymine in DNA. Hydrogen bonding between purines and pyrimidines and their stating make DNA more stable for strong genetic information. Hydrogen bonding between the purine and pyrimidine, purines and pyrimidines, hydrogen bonding, right? So that is making and the stating makes the more stable for genetic information carrying. Okay, so DNA is capable of undergoing slow mutation required for genetic material. It goes slowly, not fast as we observe in case of the RNA as genetic material. Okay, the evolution is not fast and variation is not so fast as we observe. Okay, so genetic is capable of undergoing slow mutation required for genetic material, and it has power of repairing also. If there is some kind of damages, okay, so it has got the power of repairing also. So DNA is more stable. Both DNA and RNA involves in genetic expression because RNA can directly code for the synthesis of protein and hence can easily express the characters. Right? So that was both DNA and RNA involved in genetic expression because RNA can directly code for the synthesis of proteins. So both. But in the case of this expression of the that the information contained in the DNA, okay, as far as expression is concerned, both DNA and RNA are involved in genetic expression because RNA can directly code for the synthesis of protein, hence can easily express the characters. Okay, so DNA is transcribed into RNA, and then the information carried from the DNA to RNA is then translated into the protein. So that's what it's called. First the replication, the transcription, then the RNA is formed, then translation, then there is the formation of protein. Okay, so DNA is dependent on RNA for synthesis of protein. It's not only DNA that are directly going to produce protein, but the passing on the information to the RNA for translation and then synthesis of protein. So protein synthesis machinery involves around RNA. Okay. DNA being more stable is preferred for storage of genetic information, for storing purposes. Okay. For translation, RNA is playing a very good role. Okay, more suitable role. But as far as storing of the information, okay, the coding, okay, the code that for the expression of the different characters, okay, that code that to be stored in the cell. As far as this is concerned, then DNA is more stable in storing the code or genetic information. Okay, so here you can say that RNA is see. Now we go through some MCQs. Based on which experiment DNA acts as a genetic material, and this is the Gregor Johann Mendel. No, okay. Hershey and Chase experiment, yes. RNA Kornberg, no. Frederick's based on experiment DNA acts as a genetic material. It was the Hershey and Chase. It was confirmed. Although Frederick Griffith, he also showed that DNA is a genetic material, but it was not, confirmed. Okay, and scientists are not satisfactory with the answer. From the experiment of this, the Griffith's experiment. Okay. Now, which base pair is present in RNA instead of thymine? Guanine, then thymine, or cytosine, or uracil. So it is the uracil instead of the thymine in case of RNA. Okay, guanine, thymine, cytosines are there. Okay, in both RNA, but instead of thymine in case of RNA, it is uracil, not thymine. Okay, so that is the difference. DNA is dependent on DAS for synthesis of proteins, dependent on RNA. Okay, it has the information. Okay, but it has to pass on to RNA for synthesis of protein. Okay, so RNA is the DNA is dependent on RNA, right? Now this was all about the what we call this, the search, when we study about the search of genetic to know which is the actual genetic material. Okay, how the different experiments were conducted by different scientists, and how the discovery of genetic material was done, and how the DNA as the genetic material was discovered. Okay, this was all about, you can say, as a history of the discovery of genetic material, and we study about the experiment of Griffith and the experiment of the Hershey and Chase. Okay, and which proves that DNA is a genetic material. Okay, so this was all about the search for genetic material. This is one of the very important topic for NEET, and so many questions. I can say the questions are definitely asked from this part. This is one of the important topic. So thank you for joining and listening today. Tomorrow again we will meet at the same time with another topic. Till then, goodbye. Take care.